Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us the 146th seminar, and I call it a Max Planck Institute for Terrestrial Microbiology Day. I came back and stopped by my St. Louis home after my 2 weeks of trips to Germany, Austria, Czech Republic, Hungary, Boston, and Delaware. It was great to meet many of new people and my colleagues, including today's speaker, Tobias Erb. I will embark on another 4-day trip to NIH and J. Craig Venter Institute in Rockville to give 2 talks. Okay, it is my tremendous honor to introduce very briefly, I'm not sure I can do that very briefly, our pioneer speaker, Professor Tobias Erb. He does not need any introduction because we all know him. He is a professor at Max Planck Institute for Terrestrial Microbiology. He has demonstrated a new way of carbon fixation using multiple steps of enzymatic reaction cycle, a great achievement, and I met him many years ago at Synthetic Biology Gordon Research Conference, and then I visited his institute to give a talk last year. As I mentioned, we met again roughly 2 weeks ago in Brno, Czech Republic, where both of us attended an European Synthetic Biology Conference where he gave an excellent amazing invited talk, and I was blown away by again, of course, by him or his talk. Tobias, thank you so much for your amazing contribution to scientific community for very important topic that is, of course, uh, greenhouse gas problem or climate crisis, and the virtual podium is all yours now. Thank you again.
Thank you. And I would like to thank you for bringing this community together and for having this fantastic series in which we feature maybe old, established, I would not say farts, but people like me, and then take a look at the younger generation just waiting in line like Jaya is doing. So, we all stand on the shoulders of giants, and I hope to be a dot in between connecting the past with the future. For this 2 minutes I do have, maybe 5 minutes, I want to give you a quick motivation, what we're doing in the lab. And I think this falls into the realm of thinking about global challenges and how new technologies, in particular synthetic biology, can actually contribute to bright solutions to these challenges. And in our specific research, and I hope you can see the slides already, it is really about how we can use synthetic biology to rethink, redesign, eventually reconstruct the operating system of photosynthesis of the global carbon cycle. Well, and why are we interested in the global carbon cycle? Well, at the end of the day, it's really about this small molecule here that you see, CO2, carbon dioxide, which is on the one hand an important greenhouse gas, and which puts a tremendous challenge for us, for this planet, but also for us as humankind, right? And at the same time, this greenhouse gas is also not only a challenge, but also a chance, because in principle, it's ubiquitous carbon source. And we as a society, and especially we as scientists, should be able to capture and convert this molecule in a sustainable fashion. Now, if you talk about sustainability, about carbon capture, we can of course think about human-made technologies. And that's just one example. That is a Mammoth project. It's in Iceland. It's a technology where you can really filter CO2 through chemical physical methods. However, that's just a technology that's still in its infancies. It's very inefficient, it's very energy intense, and it's actually at very low scale. And if you really think about carbon capture and you think about scale, at the end of the day, it's biology that really can capture gigatons of CO2 per year. And that is what drives the global carbon cycle. And that is what we should be thinking about if having technologies that we can use already these days. Now, if you think about biological carbon capture, the focus is mostly on plants. That's also okay. They fix around about 200 gigatons CO2 per year. But the one thing with plants is they use a single solution. It's called the Calvin-Benson-Besson cycle. So that's photosynthesis 101. And it's driven by a single enzyme, ribulose 1,5-bisphosphate, carboxylase, oxygenase, or short Rubisco. That is probably the most abundant enzyme on Earth. And it's the entry point of CO2 into the plant metabolism. Now, there's two things that you need to take into consideration. One thing is that photosynthesis and Rubisco is a slow enzyme. So if you have a perfect plant, a perfect condition, agricultural setup, you feed the plant all the nutrients, it will be Rubisco that becomes one of the limiting bottlenecks because the enzyme takes only 5 to 2 CO2 molecules per second. And there's another challenge with the enzyme. It's actually very sloppy, which means it sometimes, actually not sometimes, every fifth time, it confuses oxygen with CO2, which again also lowers effectivity and also photosynthetic yield. And so the one thing I have learned, and that many people have learned, is the enzyme is trapped, evolutionarily trapped in a Pareto optimum, where you cannot make the enzyme faster without making it less specific, or you cannot make the enzyme more specific without making it slower. So in a way, it's a bottleneck in photosynthesis, it's 3 billion years of evolution, and you cannot necessarily improve it. And so this is why I think it's a good solution, but maybe not something that we can apply and use for CO2 fixation on a grand scale, because that is a biological bottleneck. Now you can look into other solutions, right? I talked about plants, and you actually will see, and that's a work we've done with Andrew Hansen. He's actually first author on this kind of small review. There's even more solutions out in nature. So biology has been creative. There's microbes that can also fix CO2, sometimes even better and faster than plant photosynthesis. And this shows you just there are 7 other solutions on this planet. And as a biologist, I'm really amazed by all this diversity. So apparently biology has explored different ways. But then as a synthetic biologist, I look at the very same landscape, at the solution space, and I think, there's even some space left that nature has not explored. And there's an opportunity for us as human beings, for us as synthetic biologists, to actually try out this new wave with synthetic biology. And I want to make one important note here. This is not metabolic engineering where you take an existing solution like the Calvin cycle or the 3-hydroxypropionate bicycle and you try to improve it. This is really coming up with complete new solutions, building them from scratch in a dedicated fashion. And that is really synthetic biology at its heart. One example is shown here. I just want to flash this. You already mentioned it. It's one of our first principles, the so-called catch cycle, where we built a non- or human-made CO2 fixation cycle out of 17 different enzymes that's actually more efficient than natural photosynthesis in 2 ways. It can turn much, much faster, and it uses only half of the ATP, of the energy that photosynthesis needs. And so I'm coming to the end already of our, what we really would like to do is, on the one hand, we'd like to understand this biological diversity. We want to understand how biology fixes CO2, and then we want to build a solution that is eventually better. And this is a little bit like you see here. Think about how we humans ended up flying. We actually studied biology first. Airplanes were built and modeled after birds. But of course, airplanes have wings, but they don't flap, and they don't have feathers, but they still use the same principles. And that's exactly what we like to do. Understanding photosynthesis to that level that we think about new to nature solutions that are better than those that nature has evolved. And so what we really do in the lab is this dichotomy, this 2-pronged approach. On the one hand, studying biological CO2 fixation from the basic atomic parts, how the cell and the enzymes fix the CO2 to the cellular level, so the pathways involved. And then we use synthetic biology to design new to nature pathways. First, let's say in a theoretical phase. Then we find the individual parts, the enzymes that we need to kind of construct a first cycle, a first operating system, a first pathway in vitro. Then we optimize the system through several rounds. And then we move it into a cellular context. And this could be natural cell, and it could be artificial cell. And if we are lucky enough, some of these solutions might work, and we would be happy to move them further into higher organisms, for instance, chloroplast, Chlamydomonas, it's an organism we work with, but eventually also plants. And so you can see synthetic biology really across all levels. This requires a lot of motivated people in the team, a lot of nationalities, a lot of different backgrounds, and a lot of scientific backgrounds. So from biochemists, as you are, Jaya, to structural biologists, even physicists, sometimes material scientists, and molecular geneticists. And I think with that, without any further ado, I think Jaya is the next speaker, and she actually talks already about plans and implementation. So she's a bit ahead of us, and I think she has great ideas, and I'm really excited to talk to you. I just want to say, whenever you want to join us or the Max Planck Society, there's a lot of opportunity in this space, and we just need all people to work on this problem. And our solution may be part of a solution, but there might be other solutions out there, and Jaya will talk about other ways to fix carbon dioxide more efficiently in a couple of minutes. Thank you so much for giving me the opportunity to introduce Jaya. Thank you so much, Tasek, for this fantastic seminar. I stop here, otherwise I'm running too late. Thank you.
So fantastic talk as always. I mean, this is a very important problem, and then you are approaching the problem with very unique and also very innovative, solution aspect. I normally do not entertain a question, but I have one question that probably relevant to both of you. So let's kind of deal with this one very quickly and then move on to the Jaya talk. The question is coming from the Canyon. Is there still significant interest in trying to evolve Rubisco for this purpose, or have researchers mostly moved on? I know the answer, but I want to hear from you.
If I go first, I think with protein engineering and protein design, you can approach it from a different point of view. I think the enzyme itself, as it is now, the Xen enzyme is hard. So we need to use other methods. And as a matter of fact, we are also working on this together with designers. But let's say trying to engineer the enzyme in a sense, you want to evolve it as it is. That's harder. You need to think about other ways. Happy to chat. Happy for next seminar.
Sure. Okay. I believe Jaya also probably answered some of the question about that one, but let's move on to Jaya's introduction. And thank you again, Toby. It's excellent talk as always. I love your vision. I hope to you contribute to the society more, potentially solving, of course, climate crisis together. And as you mentioned, that should be solved by a lot of people. Okay. Now, the main speaker of today with a longer introduction, Professor Jaya Joshi joined University of British Columbia, Vancouver, Canada as an assistant professor in the winter of 2023. She just spent 1 year, I guess. So congratulations again. Her research program harness the power of synthetic biology and microbial metabolic engineering with tailor-made catalysts to develop novel methods for converting biomass-derived feedstocks into value-added products, in short, turning trash into treasure, which is important goal and relevant to what Toby is eventually seeking in terms of applications, although the starting feedstocks or points are, a little bit different, I guess. Her research walks through enzyme design space, exploring fitness landscapes for radical metabolic engineering design, guided by her aim of making sustainable carbon farming a reality by 2050. Recently, we met at a conference and will meet again in December at the first Synthetic Biology Young Speakers Series or SynBYSS International Conference in Hawaii, where she will give her contributed talk. Jaya, thank you so much for your time today, and please take it away. All yours now. Thank you.
Thank you so much. I really appreciate giving me this opportunity. I'll try to share my slides first so that it's easy. Can you see my slides now?
That's coming up. Yes,
It's
it's,
good?
yep,
Okay.
beautiful.
So first of all, thank you so much for this opportunity. I always felt when I was kind of a young SynBYSS, especially during the COVID time, and I really needed something which came up just like this series, which actually motivates you. But I'm also very thankful that today we have Dr. Toby Erb. I personally felt like my journey started with a interesting talk by Dr. Aaron Bar-Evan, and when he talked about Toby, and then he was giving me inspiration. Jaya, you can do it. See, Toby has that kind of a vision, so you can see that he's trying for it, and you can do it. So that's what I would say. That thank you for giving me opportunity with Dr. Erb. So I would like to start my talk saying that I'm going to talk about the continuous directed evolution. And the idea is that we are talking about some good enough enzymes and then how to make them better enzymes. So right now, as Dr. Moon said, that I'm a faculty of forestry in UBC, and then I'm associated with the Bioproduct Institute, and my lab works as the Biocycle Lab. So before even going to this, I would like to do an land acknowledgement that here at UBC, we would like to acknowledge that we are gathered today on the traditional, ancestral, and unceded territory of Gold Salish people, including the territories of Musqueam and Squamish nations. And we are very thankful to these people that we are able to conduct our research in this beautiful campus of UBC. So before even going further, I would like to say that who I am. So actually, I really like this quote. Again, one researcher from Max Planck Institute inspired me with this quote. He said that, Jaya, no man ever steps in the same river twice, for it's not the same river, and he's not the same man. And it actually applies to me. So I come from a very small town somewhere in India near Himalayan region. The specialty of this town was that girl education was not very common. So that's why I'm a very enthusiastically saying that I feel that women should take more part in STEM activities, and especially with the synthetic biology, we are seeing this coming along. So then I moved here to Canada, and then from here I moved to Florida to do my postdoctoral research with Dr. Andrew Hansen. And during COVID time, due to some family problems, we moved back to Canada, and then I moved to Montreal working with Dr. Vincent Martin, another synthetic biologist. So what I want to show here that I have almost moved like 13 cities, 3 different countries, and almost 2 continents now to reach. And that's where I feel like that's what I learned about the diversity, and then diversity among organisms and how you can harness it. So right now I'm a biochemist who eventually turned into synthetic biologist, but still I always feel about the enzymes, and then I'm interested in finding those just the good enough enzymes which I can use it and make them better so that we can capture some more carbon and then focus on the carbon farming by 2050. So going along, I think just to quickly tell you that I have done 2 postdoctoral fellowship, and both were in synthetic biology. First one was more on the enzyme evolution, while second one I learned more about the yeast metabolic engineering, so that how you can use this better enzymes and turn into some bioproducts. Before this, I have worked with the Agriculture and Agri-Food Canada, and then I worked on the plant metabolic engineering kind of project. For today's talk, I will be more focusing on my directed evolution of enzyme, and then also just introducing you with this project because I was just there for a few months, or I would say just for 1 year. So that's why this is a very short talk for this project. So First of all, I would like to really thank my team, which helped me to first understand what is directed evolution, and then I continued with the continuous directed evolution. This was not only done in Dr. Andrew Hansen's lab, but there were many people involved from University of Florida, as well as from University of California, Dr. Chang Liu. But then I really want to thank my sincere thanks to late Dr. Aaron Barr-Evan, who made sure in one Gordon conference that I pursue my interest in this area. And then he literally suggested me, Jaya, this is the time you can do it. So I feel very thankful that I got his guidance during that Gordon Research Conference. So going further, just not delaying it, that this part of my talk is called as a replaying the tape of life, and how we did it in my postdoctoral fellowship was playing with 3Ds, how to discover new to nature enzymes, how to design it, and then how to evolve them with the directed evolution. And this was funded with the NSF and then Department of Energy. So just imagine that there is this world, but then in this world, we are actually giving that we will be having a total bird population by 7.7 billion. And in 2050, we have to feed 10 billion people. And not only this, you have to improve your food production must be by 70%. And on top of it, we are wasting 30% of our food produce. And also, we are thinking that by 2050, we have to reach net zero CO2 emission. So imagine that's what we are leaving for our kids. Climate crisis, on top of that, the food production must be increased and waste should be decreased. So what as a plant scientist, we always think as a solution that you have to improve photosynthesis, you have to improve the yield, and you have to find sustainable solution for the waste. So in short, what we are looking to bring carbon revolution by 2050. But then if we are thinking about the carbon revolution, first we have to think about carbon budget. So carbon budget in terms of, I'm just bringing this concept for plants right now, but you can apply it to microbes also. So imagine you have 100 gross photosynthesis that you are fixing in a plant. And imagine how much plant uses it for itself. So if this is a pie, that 50% of it is used in plant respiration. Only 20% is used as the high harvestable yield, and 25% is unharvestable biomass. So you can see this, that 50% plant is using for themselves, 25% we are getting, and 25% is the unharvestable biomass after 100 gross photosynthesis. So this is your pizza for a party. Now we are thinking that how about the other orange part of it? In this pie, you will see that actually 50% out of this 50%, 25% plant uses for their maintenance processes. So what are these maintenance processes? The maintenance processes includes maintaining ion concentrations, nucleic acid turnovers, or even synthesizing new proteins, or which we call it protein turnovers. And imagine that I'm thinking that in soon we have to feed a lot of people. That means we might have to order big pizza. And that's what a lot of people are doing. They are thinking that to feed more people, that one solution is order bigger pizza. That means you increase photosynthesis. That means you will have a lot of harvestable. The harvestable is the same, but then you have enough to feed people. But we are thinking in a different way. We are thinking, why not reduce the maintenance cost? That means just change the size of the pizza. That means if you reduce the maintenance cost, and also if you would like to, and if we are successful in increasing photosynthesis, and if you can combine the strategy, you will feed more number of people. And that's what is the goal, that 70% increase in our food production. So coming to the protein turnover, we are looking at why protein turnover is important. So when we looked into this stat of it, we realized to synthesize one residue or to break down one residue, it requires 6 ADP. So that means it is very expensive. So you can imagine that if I can reduce the turnover, I will still be able to decrease the respiration maintenance processes, but increase the harvestable yield. And that's why we can combine this strategy and feed more number of people. You might be thinking that why Jaya is saying that what we will do with this increased biomass. So imagine this, I was just reading 2020 McKinsey's estimated report, and according to them, 60% of the global economy physical inputs could be made using synthetic biology. So which is resulting in a direct economic benefit of at least 1.7 US trillion between 2030 and 2040. So imagine if I have this extra biomass carbon, what I could do. I could solve 60% of global economic problems, or maybe we can put some physical inputs. And then if we can install new to nature designer pathways or biopolymers or something like that, we use these new biocarbons to construct new to nature biopolymers or even a new to nature sinks, which we could use in the future. So that's where this extra biomass carbon could be used by the plant or by us by creating new sinks. So coming to this one, I would always say that that's why I always feel that this is the time when we have to think about creating extra biomass carbon and then using it and installing new biocarbon into some biopolymers or bioproducts. So when I started my postdoctoral work with Andrew Hansen, a study published by our collaborator, Dr. Harvey Miller, that they analyzed a degradation rate of 288 orthorhombeous barley and Arabidopsis protein. In this graph, x-axis is the degradation rate in barley, and y-axis is the degradation rate of Arabidopsis genes. So what we saw that two proteins turn out to be having very high turnover. One, which we already heard from Dr. Erb, that one was infamously famous D1 photosystem 2 protein, and then second one was the thiazole synthase. This thiazole synthase, which is red in color here, is the highest turnover rate in Arabidopsis and barley, with half-life between 2 to 10 only. So you can imagine how quickly this enzyme dies in a plant and how you have to recreate this enzyme again and again. So this is Arabidopsis and barley leaves they studied, and there was the fastest turnover rate enzyme was Thi4. This Thi4 is known as a suicide enzyme. It makes thymine thiazole moiety, which was published by Begley in East. And then Begley's lab also concluded that this is a suicide enzyme. And what I mean by suicide enzyme, I'll explain you here. So how Thi4 is a suicide enzyme? This is a Thi4 wild type enzyme. So what it does, to make a thiazole moiety, it needs a sulfur. Instead of taking it from around, what this enzyme does, it actually gives its own cysteine as a sulfur source. So, and then finally, this cysteine is actually turned into dihydroalanine. That means now, after one round of reaction, this enzyme is dead. It can't do the reaction because cysteine is now converted to dihydroalanine. So that means it does one reaction. So as a biochemist, I would even say this is not a true catalyst because it just does one reaction. Catalyst definition is the catalyst should do multiple rounds of reactions. So that's what's the case. But if as a synthetic biologist, I think look at the cost of this one, it is actually almost 2 to 12% of total maintenance respiration. So you can imagine that this one enzyme uses 2 to 12% of total maintenance respiration. And then if I can improve it with my synthetic biology tools, then what I am thinking that it should be able to take sulfide from the environment and then instead, so that means no cysteine is converting to hydroalanine. That means still my enzyme is alive and that it can do another round of reaction or more round of reactions. So if you put this one, how much maintenance respiration it's gonna cost. So if we just cut the 5% respiration with just improving this one enzymes, we will be gaining several percent of biomass gain. So Dr. Henson's lab earlier actually projected this, that if this is your wild type Thai4, and if you have a low respiration non-suicidal Thai4, you are actually increasing 4% of total biomass gain. And if you go to a breeder and ask them, is it good? They will be saying it's wonderful, because this is large in breeding context. 4% yield increase in biomass is big. And interestingly, this concept could be applied to any crop, because they studied in Arabidopsis, barley, and then Thai4 is same in most of the crops. So you can imagine what we could do with this suicide enzyme. So what we did, that we first looked into whether these few plants have this non-suicidal Thai4. It's just that plant biologist mindset that maybe some plants have it. But then when we looked at it, we realized that actually few plants like wheat, oats, barley, they had these ones, but they were only localized in the grains area. And you know why? Because there is a hypoxic conditions. That means there is no oxygen condition, and it only appears in the grain region. And when we actually expressed in E. coli, where this green one is Arabidopsis Thai4 just as a control, you can see that we were able to express them, but then they exist, but then they are seed specific and they are very low activity in E. coli compared to some prokaryotic catalyst, which was reported by Bagley. So that's where we started looking outside of the kingdom. That means we started looking outside of the plant kingdom, and then we started doing parts prospecting. That means finding new Thai4 replacements. So Begley already, when he reported this as a suicidal Thai4, Begley's group already said that there are some non-suicidal Thai4s, which are 2 catalysts. That means they do more than 1 round of reactions. They reported them in a very hyperthermophilic anaerobes. And according to them, they require high sulfide environment. That's why they need their cysteine. And in this case, cysteine was replaced with the histidine in case of active sites. So what we did, we started looking that, see, this is Arabidopsis cysteine and this histidine. Do we have more such things which we can bring it to lab and then work with them? And then if possible, one day we can put it back in the plant and then replace the Thai4. So we started doing the comparative genomics, where we were looking for other non-cysteine Thai4s and whether they are catalytically active and can we evolve them. So comparative genomics study actually revealed that yes, they exist. And we did for this is called the sequence similarity networks. For the sequence similarity networks, each point is 1 sequence. And when we just put them according to their sequence, and what we realize that they are clustering with the similar sequences. That means they exist, but they might be having some common property. That's why they are clustering very differently. And that's what happened, that we started choosing for the test. And just to give you one idea, that see, this one, the Methanococcus cluster, was actually reported by Begley's group, that they are those ones which are in a hyperthermophilic condition, are found in a very high sulfur conditions. So you can see our research was going good. But just because we are doing, I was the first one doing this, so I'm like, okay, I will choose few of them, and I started working with them, expressing them whether they actually are the true catalyst or not, and doing more biochemical analysis with them. So with this comparative genomics, we realized that they do exist in archaea and bacteria. They are facultative aerobes, or some are even low sulfide environment. So this gives you idea that in these ones, some of them were cysteine was replaced with the histidine, and in few one, this was not even histidine. It was replaced by other amino acids. So what we decided, that can such Thai4 be evolved to function in plants? And exactly how Dr. Erb said that we have to look outside the kingdom and then find these synthetic solutions. So we started looking that, okay, can we evolve them? But I would say that first you have to look at the structure. So we decided to look at the yeast structure, which is a suicidal Thai4. And then we looked at the methanococcus structure, which was again published by Begley's group. And we were like, okay, there is a difference in the active side because there is histidine. And this one is, in this case, we started putting our, which we actually, from the sequence similarity network, this was one candidate, TA-Thai4, and then we saw that this has actually the active cleft of our candidate resembles very much with the non-cysteine containing Thai4. So we were like, okay, structurally, we are also thinking that this one which we are thinking as a candidate are very much matching with the description of a non-suicidal Thai4. So that's why we were like, okay, let's go behind them. So what we did, I'll go a little bit quicker here. That's just to explain that once you know these parts, you have to think about how you are going to bring these facultative aerobes enzymes to plant. So first requirement is they should functionally in air because that's what we want for plants at higher sulfide maybe. So that's why we decided to screen the E. coli knockout strain where this Thai-G is equal to Thai-4. So that's where we decided to go behind this E. coli thiazole oxotroph. And then we started putting them in aerobic and anaerobic conditions with some cysteine to increase internal sulfide. And then that's where this was the idea that in anaerobic conditions, this one is empty vector. And against that, we saw, yes, in anaerobic condition, few of them were actually growing or complementing in the presence of the cysteine. But we are not very interested in anaerobic because for plants, we are thinking about aerobic conditions. So the second experiment was in aerobic conditions. And then we started looking for the same thing where we were putting thymine as a control positive. And then once we started looking what happens in aerobic condition, that's what we saw that yes, in play when there is a cysteine, they grow in aerobic condition a little bit. But we were very happy to see that even without cysteine, they were able to grow a little bit. So this is the hope for a synthetic biologist that there is some activity already exists and only thing we have to do them, we have to make them better. So, but then the second one is for the directed evolution experiments, we are thinking that we do have candidate, but then the second requirement is there should be some growth in liquid media because that's where it's aerobic conditions, you're going to grow them, you're going to do some kind of a passaging. So that's why we decided to let's go and take few of these ones and then started growing them. So, for 24 hours, we didn't see anything compared to the empty vector. And this is just the wild type as a control. And then we were like, it's not growing. But I think for the evolution experiment, you have to have some patience. And that's what happened. That after almost like 2 days, we started looking some growth here. And then we were like, okay, we found the growth, but still this is not very good. So that means what we are thinking that we could evolve them to make these Thai4s better, which are non-suicidal Thai4. And the second thing we are looking to select for growth rate. That's what we were looking because thymine is directly, you can put it in the media or remove from the media. And then the idea was that slowly we will remove the cysteines as a selection pressure. And then eventually, once they are evolved to grow, and then without cysteine in it, we will be able to transfer them to plant, or we can engineer the plant cysteine, whatever kind of the residue change we see in our improved Thai4. So this was whole idea, but I would always say this is never easy. So that's why I thought that how to do this enzyme designing. And then this concept came that enzyme space is vast. And I really like this slide from Andrew, which I'm presenting here, that he always tells us that SynBio and enzyme design space, when you think about it, the enzyme design space is mathematical concept, a space containing all possible design. That means it's very, very vast. And then we started thinking that, okay, the possible protein design space is enormously vast. So just if you have a 300 residue protein, imagine you can have these many combinations. But of course, they all don't exist. And if you just compare with the 10 to the power 80 atoms in universe, actually with protein, you have this much more possibility, even with the atoms in the universe. So you can imagine that opportunity is vast and the space is unimaginably vast. So that this comes to the concept that why plants didn't do it? That's why they didn't do it, because they has explored very little of it. That's why few enzymes are still not doing that good. And that's what I always feel, that Rubisco, we can make it a little bit better because it is not yet fully explored yet. And there is a very few of the possible enzymes and pathway actually exist. And Dr. Erb already identified this. And again, I always feel that this example given by Dr. Andrew Hansen was amazing. He said that in this paper, when they looked at the glycolysis and they found in life only 2 kind of pathway exist, and we know that. But then when they looked around synthetically, how many possibility, and they found 12,000. So imagine that if glucose to pyruvate, you are thinking of 1, 2, 3, and then synthetically, there are many more possibility. So that's why you have to think enzyme design space as an unimaginably vast area, and then you have to think through it. So coming to make how you can use enzyme evolution to make super catalyst, actually it goes back to one of the SynBio's most iconic achievement, and which was also the first talk of the SynBYSS series, was Dr. Francis Arnold, who won 2018 Nobel Prize for the enzyme evolution. And that's where you see that repeated round of mutagenesis and selection to find new activity. And so if suppose you have one protein which you want to mutate, the mutated version you select, you go again and again, and then you are actually selecting the best. Imagine if you can do it in vivo without using your hands and without actually going back and back again in the lab. And that's what continuous directed evolution does. So like I said, that you have A, which is actually your protein, which you are thinking. So there is a chance that you can make A better like B. And there is also a chance that you will go through a fitness valley where you might not be getting this. But with the correct selection pressure and the sequence space, you will find sometime a very good global maxima, which is a C. So that's why this is a vast space, but there is a possibility that with repeated route of mutagenesis and selection, you will be able to find a new activity here, or I would say a new scale of activity. So you might have heard about the rational redesign quite a bit, which people have done quite a bit, that suppose rational redesign is more like this, that you have Arabidopsis thai4, I just changed C to histidine, just what Begley's group has reported. But then I thought, what if one mutation is not enough? What if we need more mutation to get the activity to the global maxima? And then second is, you don't know which residue to replace. In this case, yes, we knew a lot of things, but suppose we have no idea. So that's what in that HI4, we started thinking about the directed evolution, not the rational design. So doing some random mutation and screening. So I would say, actually, directed evolution is a kind of game of Plinko, that you start with one, you go with a bunch of them, and ultimately you hit the jackpot activity. So for this, we were looking for the rate of mutations, selection pressures, and need a platform to evolve. And luckily, at that time, Dr. Changdu's group has reported OrthoRep. I would definitely ask you guys to follow these papers because this is amazing system to go with. And then there is another system, which is a MutaT7, which is again, this is E. coli-based system, but we went ahead with our yeast system, which was available. And then he has given us this scalable automated continuous evolution platform. So I would not go deep in the OrthoRep system because there are very many young investigators which are using this. But then this has the orthogonal system, means gene of interest is sitting outside, and then we have a high mutation rate polymerase, which is going to be doing the errors, and that's why you will have a high error rate. And then finally, you can do the evolution. But for our purpose, we had to customize these plasmid, and that's what we did. And ultimately, what we ended up doing is a proof of concept study, and then I left here after working with this system, and then a few of our postdoc continued with this, and they were ultimately able to find few of the mutation now, which they are still working, is associated with this aerobic one. So with these mutation, they are doing it. So I'm very thankful that they are continuing this work. So just quickly go to my second one, which was I moved to Dr. Vincent Martin lab. Here, my aim was to understand synthetic biology in a larger scale, not just go in a smaller scale, but big one. So I started working with the how to create green cell factories here by construction and editing. And this was the team. I was very thankful to learn with this team, the metabolic engineering, all the beautiful things about the non-traditional yeast. And that's why our idea was that a lot of dairy industry threw a lot of cheese product in the I would say they go in the drainage, which is not good. It's organic produce. So that's why, what if we can convert this lactose to fumaric acid or some other organic acid, which could be used in our food additives and all. So this was the bioprocess development for the valorization. And then, like I said, this was done in the large scale. And this what I learned here was this, that you do have to have automation with the synthetic biology, because that's where I found that biofoundries are key for SynBio. And I'm very happy that SynBio, there is a network of these biofoundries, and we can use it in globally, and definitely it's very useful. So coming to very quickly, for 5 minutes, I'll quickly go through with my lab what we do here, because I moved to UBC last year, and then this is my this just completed 1 year. So what we are doing here, we are replaying the tape of life with 3 E's here, which is enzyme evolution, editing, and engineering. My lab is called as a biocycle lab, and then we are thinking about reduce, recreate, and recycle. And why we are doing this? We are thinking about the food, feed, and fuel, what I said in my first slide. And we are bringing the revolution by doing some enzyme evolution. So to reduce the carbon loss, we are going actually behind some of the enzymes which are not good enough. They follow this song very well. Live hard, die young. And then in my previous paper, we actually calculated their metabolic flux rate and enzyme replacement, and we realized that their enzyme actually die very early. They have a high risk reactions, and then and the trend is very similar if you just look at into bacteria or yeast or Arabidopsis. So my idea here is to find these live hard, die young enzymes, and then with the help of continuous directed evolution, we will be non-essential residues to prevent damage. So we will be replacing these residues. So we are actually improving quite a bit of enzymes in our lab. The second platform, so not going to the second platform, but quickly that, like I said, that last time I showed you this, and you might be thinking, Jaya, you chose few of them. If you lost the good ones? And that's what I felt. So this is why my lab actually have a platforms where we are using machine learning, which will be able to predict few of these candidates in the beginning, so that we are not losing the good ones. So we are also developing a machine learning platform by developing some CNNs for protein designs. The second platform, this first platform is for green biotechnology, and then idea is same, to create new carbon sinks in plants for fuels, food, feed, and traits. The second one is to recreate cell factories. Like I said, we create the editing platform here, and then that's why the idea is, like I said, that what if we can build and install a whole set of designer enzymes? So like I showed you that for Thai4, we have to do directed evolution because we really need this enzyme to do much better. But maybe few enzymes just need to do the rational redesign. So if we can combine these activities, then we can build and reinstall these ones. So here we are helping here with the enzyme evolution and editing for wide biotechnology. So we do work with some industries here where we are thinking how we can contribute their cosmetic, textile, and food additives, or even the plastic upcycling. And last but not the least, this is my favorite one, how to recycle feedstock waste, how to enter in this whole circular economy concept. So my motto is that waste is not the waste until we waste it. And that's why we always think about how to valorize the feedstock and how to enter in circular bioeconomy. So we think about how to bring from sink to shelves or trash to treasure. For that, we have two projects. One is to grow forest products in Petri dish. That means we are actually growing next sustainable cellulose supply. So bacterial cellulose is the one we are targeting right now, but we are using feedstock to get the bacterial cellulose supply, and then we are using quite a bit of metabolic engineering principles here. The second one, this is with the bacterial stain. The another one which we are just starting is a plastic upcycling. So we are whatever plastics, if we can break into smaller pieces, and how to make them as a treasure, not a trash, because Vancouver has a lot of ocean pollution just because of these microplastics. For this, we are using non-conventional yeast. So just the final one, which I always said, that replaying the tape of life with CD, that's what I want to do. That I was very fortunate that I got the help of these funding agencies all the time, but majorly these three people who shaped my career as a young investigator. But then because of their network, it literally grew with the more and more number of people. And I just want to keep growing this network, which literally Dr. Erb showed you that how you can find the different synthetic networks also in this case. So with this one, I would say that that my whole synthetic biology journey actually started with Dr. Erb's this CTEC cycle. And then I saw that they have a 17 different enzymes from 9 different organisms. My idea is to how to bring back to the life. So that's why I would say that now I could see that many researchers can come together and make it possible. So with this, I would say that I said that we have to do 2050 carbon farming, but that you have to do with SynBYSS in 2050. You have to start now if you are thinking about 2050. With this, I would say we don't want to leave our world with these kind of problems. We want to leave this for our children. So that's why we have to make some contributions now. And with this, I would like to thank my previous teams for bringing this synthetic biology as a revolution. And like Dr. Moon always said, as a synthetic biology, that we have to think through it. So I always feel it that this is a very important concept. And with this, I would leave you with my previous acknowledgement. And also, this is my small lab right now. And then we are thinking of growing it. So we have 3 graduate students. So we are looking for SynBio enthusiasts in the UBC campus. So if any one of you are really interested, I would definitely like to get your help to bring this enzyme evolution for revolution. So I'll stop now here. I have already taken 5 more minutes. I'm very sorry for that. It's just that I thought that this will be good to bring my lab idea here also, so that we get the more SynBio enthusiasts. So thank you very much for this opportunity. I will stop share now and over to you.
Yeah.
Yeah.
So fantastic talk. This is a lovely talk and then lovely vision. Let me see whether we have question. And Toby need to go to his kid. I'm not sure he back now, but let me start with my question. So you mentioned evolution. I believe evolution is a very important aspect of the life, of course. And I use a lot, evolution, directed evolution because my academic grandma also Francis Arnold.
Yeah.
The question I have is, we haven't explored the space of
Yeah.
mutation, but oftentimes when we use the directed evolution, we suffer from false positive, false negative. That is sometimes kill the motivation and enthusiasm from my student and postdoc. So how we gonna avoid those kind of force, or cheaters when it comes to implementing directive evolution?
No, this is a very good question. And I would definitely say when I started thinking about the directed evolution, I also thought exactly same, that I might be getting more false negative and some even false positive, because then I'm be like, okay, these are not good. I'm not using it. But what if they actually work better in the another kind of selecting pressure? But I realized over the years using these systems that we are getting actually better and better with our systems. So the OrthoRep system, which actually was a 2013 time when Dr. Liu's group actually published, and I saw yesterday a paper in Science where they actually improved the system and quite a bit of offsite targets getting in. So I personally feel that with this technology and what we learned, and I again say it's all credit goes to Dr. Arnold, that she started it and she actually built it. So now people are actually exploring quite a bit of mutational system where we will be having a better, less and less off-target effect. But then second thing, which I always tell everybody, that you have to design your campaigns so well, that how you are going to look into the promoter region, how you are going to look into, how you are going to be saying like, okay, this is only happening in my gene of interest. That's why I'm seeing this growth activity. So designing your campaign is a very important part. And I think third, which is very important now, to combine this machine learning uh, concept in the beginning, not while going, because definitely we have a vast amount of data. This is very important to bring this AI power. And I feel very good when I saw that this year's Nobel Prize also went for the protein design, and just use those tools. And with those tools, it's getting better and better. Even what for the Thai-4, I would say I have found new candidate just by using existing tool. So I would say that this is going to get better. But I always say that as a biologist, we have to think through our whole campaign very well, that what tools and where we have to use this efficiently. Yeah.
Yeah, I agree with you. I also traumatized by protein engineering because when I was a graduate student, I became a robot. I'm talking about liquid handler myself, also colony picker myself. So I picked more than 40 colonies, I believe, at the time to just uh, engineer specificity of the enzyme. Even though we use uh, the computational algorithm to design the protein, but that is still 10,000 or 20,000 variant. So now I rely on a lot AI, machine learning, AlphaFold, Rosetta. That's an absolutely great tool. But the question I have is to make your enzyme super efficient or stable. Sometimes we probably rely on, I would say, completely different scaffold or new enzyme. Because I mean, I do believe somewhere in the universe, there should be another set of the living systems that probably has much more efficient. I don't want to say Rubisco, but let's say carbon fixing enzyme that's much efficient because they didn't end up local minima like we did. So to go there, I mean, we need to start from very much new space, new starting point rather than existing enzyme. And I just want to hear from you about your perspective on that point.
Yeah, no, I would say that this is a very important one, that there is a space which we didn't even explore. And I agree with it. I think for what I personally feel that having this metagenomics data, and I think Craig Venter Institute helps you to get a lot of metatranscriptomic data even to get it. I think that is personally, I feel a very good start for my plastic degrading enzymes. That's where I'm going. I'm not going just behind what we know about one or two bacteria which has the plastic degrading enzyme, because technically PETase is, we have done whatever we could do with it with directed evolution. But then there are more coming up. There are more enzymes you can find in the space. And so that's why I feel that we have to combine this whole the metagenomics data, which is actually people have done wonderful job in extreme environment and even in the oceans and all. So we have to bring that one into picture when we are talking about the new to nature enzymes, or even thinking about, oh, we will find the new enzymes. So you don't have to just sit in one kingdom or two kingdom. Actually bring back those metagenomics data. And that's why I always feel that Winter Institute has this fantastic power, but they give, and they even run those projects. So this is like what I always feel, this is a very good opportunity that as a young investigator, I always feel that this is where I would go for it, to find actually the other world of those enzymes which exist, but we don't know yet, because we are still sitting in our silos. We are looking only on those two or three enzymes. Yeah.
Absolutely. So let me check with Toby came back, or he's still picking up his kid. Toby, are you there? I guess he's picking up his kid.
He might be picking up. That's
Okay.
fine. Yeah.
Okay, so let me check whether we have question from audience other than the one we had. Let me go back to question. By the way, do you have any answer to question Canyon already asked? Is
That's
there
very
still
interesting. My student. And I think this is interesting that Rubisk, he is, but Toby said that that's what I learned when I started mostly trying to fix this other angle. I was going on the maintenance respiration, and I've seen when I was in US, everybody was targeting more on the photosynthesis side. And I realized talking to everybody that engineering Rubisco is from different angles people are thinking. Even now, they are actually creating the ancestral, looking at the ancestral part of Rubisco and how you can improve from that side. So I would say that with those new technology, new strategies to bring these enzymes, I think people started thinking, oh, we could do something better. But I would still say that people are now moving away from Rubisco a little bit more, and then finding more kind of solutions, and not just looking for Rubisco. And I think CTEC cycle was one example. You don't have to just focus on one or two, but you can have a set of enzymes to bring back your objectives and reach the goal. So I personally
I
feel that,
agree.
yeah.
okay, Toby
Yeah,
back.
can you hear me? Yeah,
Yeah,
no, I just had a hard time to unmute myself somehow on the mobile phone. This turns out to be more complicated. Good thing I'm not a mobile phone engineer, but a bioengineer.
sure. Sure.
I had really only one question. It's fantastic talk, by the way, as much as I could follow it on the bike. So what's the actual potential if you had every enzyme you could think of, maintenance and so on and so forth. So how much can you actually get out there in total, right? That
Yeah.
would be the first question. And second of all, if you make enzyme more stable, you also tune and deal around with degradation, right? And proteolysis. Would you also tap into this pool to kind of slow down degradations? Or so maybe these two questions from my side. Everything else was fantastic.
Yeah, no, Toby, that's a great question. And I would literally say that this was work which we did in COVID to literally look into kind of more than 300 metabolic enzymes in Arabidopsis, Lactobacillus, and yeast. And then we found actually 20 to 30 enzymes are on the lower CCR side. That means they die very young. That means they do a 1 to 100 or even 1 to 1,000 reaction and die. So personally, I feel if I put this whole thing that just Typhoid is like very notorious, so I'm saying 4% yield increase. But if you can combine this and improve every single of this and then create that awesome plants, which is literally designer plants, I personally will feel like we can even improve like 7 to 8%. Given that I'm not targeting right now the degradation rate, because then there is another whole angle we are dealing. Right now, I'm just trying to improve their shelf life, or I would say that a lifespan. So I'm not talking about that how much they will degrade even few, because I'm not going to be able to bring them their maximum cycle, like 10 to the power 6 cycles, no. But then if I can improve them even 5% or something, I always feel that we will be hit 8 to 9%. But I would say that that few enzymes with typhoid we have seen, it's so tricky to evolve. So we are trying all sorts of things. But then my lab actually focusing right now on cysteine synthase, which is doing much better. But then it is mostly responsible for cysteine and methionine biosynthesis. So most of the things we are seeing in more kind of a legume, we will be able to see quite a big effect rather than just in the grains. Yeah.
Yeah. And maybe a second question related to that in terms of one thing is to improve enzyme catalytic turnovers. The other would be to also move, for instance, expensive amino acids away from expensive amino acids, so lowering the amount of high maintenance / expensive amino acid content. Is that something you also consider or tried it at some point?
See, right now, because with the directed evolution platform, we are thinking that the moment we start getting these better enzyme, then we will definitely go back and analyze these amino acid residues and see that these are actually like a better deal here. And I think definitely I would like to see in my lab going in that direction. It's a really good suggestion to go in that direction.
Yeah. Well, of course, it always also, of course, in nutritional values at the same time. So it's probably two-edged sword to some extent. Yeah. But cool, really cool. So hope to see you soon in person. I'm not coming to Hawaii. I cannot fly. I need to save CO2. But, I think it would be fantastic for us. It's organizing. Okay, I have to, I think, log off. This is fine. But thanks for this great seminar, for this inspiring speech, and entire thing. Super cool seminar series. Thank you so much.
Thank you, Toby. I mean, 2 years from now, we'll have conference in European country.
Okay, that's much easier for me. I can come by Deutsche Bahn, but then it takes 2 days.
Sure. All right. So let me double check. We don't have any question anymore. Okay. We don't see more question from the audience. Okay, let's close because this is already more than 1 hour. So let me close and then we may chat more. So thank you all for joining and staying today. We'll meet again on November 21st, Thursday, the same time, the same Zoom link. That's a normal schedule. We'll have Professor Madan Babu at St. Jude Children's Research Hospital, and also Professor Brandon Debkowski at Massachusetts Institute of Technology or MIT. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I stop recording. Just give me 1 second. Just stop recording.